DNA molecule is known as deoxyribonucleic acid. It is a type of nucleic acid that's made up of ribose that is missing one oxygen molecule. One oxygen atom. Okay? So it's deoxyribonucleic acid. So a nucleic acid has a monomer known as a nucleotide. When you put a bunch of nucleotides together, it's called a polynucleotide, poly meaning many. The nucleotide is made up of three things: a sugar, a phosphate, and a base. In DNA, you have 4 bases: adenine, thymine, cytosine, and guanine. Now, I wrote this down this way, at CG. I try to remember at the Coast Guard. The A always complements T, and the C always complements the G. Okay, and I'll show you that why later on. The sugar is known as deoxyribose. In RNA, it's just ribose. Remember, this one is missing an oxygen atom. Okay, so when you come over here, this is your DNA double helix molecule. Right here, you can see this is D for deoxyribose, and then you can't really see the phosphate here. And then deoxyribose with a phosphate, deoxyribose and a phosphate. So that makes up this part here, where everyone's the same. The difference comes in when you have different bases. So these bases go in the middle, and they're like rungs on a ladder. This is a twisted ladder. And then the A goes with a T, A goes with a T, C goes with a G. So that's what you need to remember. And so this right here is one nucleotide, okay? So what happens is that this is what it looks like in a diagram form. And you can see this sugar, phosphate, sugar, phosphate, sugar, phosphate. C goes with G, C, G, A, T, C, G. That's how it works. And those are the 4 words right up there. Okay? The 2 strands are held together by hydrogen bonding, which are pretty weak. Now, a DNA molecule is what makes up a chromosome. So here's a DNA molecule, and it's wrapped again and again and again around certain proteins called nucleosomes. They're wrapped again and again and again, wrapped around really, really tightly. So a packly tight DNA molecule is known as a chromosome. They just did 2 of them. That's the sister chromatid, so this must have occurred after the S phase. And the centromere is the middle. This is the cell membrane. This is the nucleus. This is your DNA. The DNA does not leave the nucleus. It makes a copy. And just one little piece of the DNA, let's call it a gene. So this gene, which is just instructions, is made right here, and there's the copy called mRNA, messenger RNA. RNA is ribonucleic acid because it has ribose, not deoxyribose. So the RNA leaves the nuclear pore and comes out of the cytoplasm. And it connects with this little thing here called a ribosome. The RNA gives instructions, the ribosome does the work. And it tells you which amino acids come together to form a protein. Remember, a protein is a bunch of amino acids stuck together, called a polypeptide, and then it's folded to become a functional protein. When you go from DNA to RNA, it's called transcription. When you go from RNA to a polypeptide or a protein, it's called translation. So remember, transcription occurs in the nucleus because DNA is always in there, and translation occurs in the cytoplasm. So this is what a chromosome looks like. That's the centromere. These 2 are sister chromatids. This is what you're going to need to know, right? This part right here. So this is a DNA strand up here. What you're going to do is come up with another strand of DNA because it's double-stranded. So you have A goes with T, T goes with A. So you would actually write T, A, C, G, G, C, T, C, right? You get that. You go across the entire thing. When you transcribe it, now you're going to have to go from DNA to RNA. So you're transcribing. The one thing you need to remember is that RNA does not have thymine. It has uracil. So where DNA has ATCG, RNA has AUCG. So you see how that has A? So a T would go here. So what would come here? A. This is a T, so this would be an A. But now when you go to RNA, you don't put a T, you put a U. This is a G, so this goes here as C. So when you go from this C, it goes back to this G. Okay? Now, right here, I just did AUG. We're going to translate this using a table to come up with amino acids. So these are little circles to represent where the amino acids would be. The RNA tells the ribosome which amino acids to connect. So I had here AUG. Okay? So in this table here, you find AUG, methionine, which is start. It will always hit the methionine. If it doesn't start with methionine, keep going across until you find the start, and then start your polypeptide there. So you go there and you find it here. So you literally are going to write here MET. You don't have to write methionine, just write the another abbreviation. So with this one here, this is going to be CCG. So up here it's going to be GGC. So this is going to be CCG. So I go to this one and I go CCG, and it's going to be proline or PRO. So I come here and I write PRO. And you keep going across until you get to a stop. This is an example. UAA is a stop. So then you would write stop, or you can just completely stop, and that's it. Okay?